Hello, this video is about the law of diffusion and membrane transport. The law of diffusion is a law that can't be broken unless you put energy into it. And we're going to talk about the energy dynamics after I finish talking about diffusion, osmosis, and tonicity. Now, the law of diffusion is based on something called the random thermal motion of molecules. Random means that it's unpredictable. Thermal means that as it gets warmer, the motion increases. And motion means that particles are going to move and bump into each other. And when they bump into each other, they're going to ricochet off and go in another random direction. Now, where do molecules get this random thermal motion? Well, it comes from vibrations in the bonds of the molecules. So for instance, if I were a water molecule and my head were the oxygen and my hands were the hydrogen, there would be vibrations like this, and like this, and like this, and like this. And because water molecules don't have eyes to see where they're going, sometimes those vibrations would make the water molecule collide with another water molecule, and then both of them would go careening off in different directions. Now, the more concentrated particles are, like the more particles there are in a particular area, the more collisions there are going to be. And the more collisions there are, the more the particles are going to move off in a different direction. So if we look at a scenario where I have a chamber that's divided in half, and on one side I have a lot of particles. We're not worried about what kind of particles, just particles, because they all obey the law of diffusion. On the other side I have not very many particles. Now, if I'm thinking about random thermal motion in these two chambers, there's going to be many more collisions on this side because there are simply more particles. Over here, it's harder for the particles to have collisions because they're more spread out. They're more likely to miss each other. So on this side, if there is a collision between two particles, let's say these two, one will go off in that direction and one will go off in another direction. And of this, the others, they're all going in different directions. Okay, all over the place. Same thing on this side. But what happens is when I take this barrier away, most of the particles are going to end up moving in this direction because they're colliding with each other more and they're going to tend to move in that direction. Now, there is still movement in every other direction, but the net movement, that means if you look to see where the most movement or the direction of the most movement, then the net movement is in this direction. And we say that it is movement from high concentration to low concentration. So these particles will move from where they are concentrated to where they are less concentrated. You can think of particles as being a little bit like people in this sense. We don't like to be crowded. So if you're one of a lot of people on one end of a field, then there will tend to be movement away from other people so that we spread out. And particles are the same way. Now, they don't stop moving once they're evenly spread across the entire area. In fact, they still move in every direction. But what stops is the net movement. Because there's no longer a difference in concentration from one side to the other. We say that the net movement stops when equilibrium is reached. Equilibrium is when things are the same on both sides. Now, we can't get particles to move or to have net movement from low concentration to high concentration without putting in energy. And that's where we come, or that's what we come to when we get to membrane transport. Okay, so we have net movement from high concentration to low concentration. And the speed of that depends on several things. One of them, as we mentioned, is temperature. So the warmer it is, the more collisions there will be, the faster the diffusion will be. Another thing, which we also mentioned, is the concentration. Okay, so the greater the concentration difference between the two sides, the faster things will go. And let's actually say that. We're talking about the concentration difference. Okay, now what do I mean by this? What I mean is that if we have two scenarios like this one, and they have different differences in concentration, then the rate of diffusion will also be different. So let's make our two chambers. And instead of drawing dots now, I'm just going to write some numbers. So let's say that in this scenario, I have 100 particles, and on that side, we have 10 particles. And on this one, I have 100 particles, and on the other side, 50 particles. Now the difference in the concentration, which we symbolize with the Greek letter delta, that's a triangle, the difference in concentration here is 90. The difference in concentration down here is 50. So this has a steeper concentration gradient. Gradient just means the space over which the change takes place. So really you can think of this as just a hill. So if we go from 100 feet to 10 feet, that's a difference of 90 feet. That's a steep hill. If we go from 100 feet to 50 feet, that's a difference of 50 feet. It's not as steep. So this has a steeper concentration gradient, or a steeper hill for the particles to go down. And what that means is that while in both of these scenarios, particles will diffuse from high to low concentration, it's going to happen much faster here. So the steeper the concentration gradient, the faster the diffusion is going to be. Okay, next thing is the molecular weight. Which corresponds to the size of the particle. The heavier it is, the bigger it is. And as it turns out, the bigger the particles are, the slower they move. And that means that they will also diffuse more slowly. So as temperature increases, the rate of diffusion goes up. As the concentration difference increases, the rate of diffusion goes up. And as the molecular weight increases, diffusion goes down, or the rate of diffusion goes down. Now there are two other things that affect the rate of diffusion. I'm going to write them and then I'm going to demonstrate them. One of them is membrane surface area. And the other is membrane permeability. Now in the scenarios that I've just shown you, we didn't really have a membrane, we just had a line to separate the two different sides. But let's look at what happens when we have an actual membrane, like a cell membrane. Okay, so let's say I have a cell, and its job in this part of the membrane, just that one side of the membrane, is to allow the diffusion of some substance, we'll call it substance X, to into the cell across the membrane. Now, with this much membrane, there will be a certain limit on how many particles can diffuse across in a certain amount of time. But one thing that cells do is they say, all right, well, if I just had a lot more membrane, then I could transport many more particles. Those are supposed to be X's, right? The problem is that cells aren't allowed to just spread out wherever they want to. So what they do to increase the surface area is instead of having one flat membrane, the cells take the space that they have and they squeeze more membrane into it by allowing the membrane to curve like that. Now that has the same surface area as this, and therefore can allow many more particles to diffuse across quickly. We saw this when we were talking about the cell. This particular feature is called microvilli, or tiny fingers. And that increase in surface area is there to increase the rate of diffusion or transport, which we'll talk about shortly. Okay, the next thing is membrane permeability. It goes hand in hand with membrane surface area. Cells usually maximize both of these at the same time. But let's take our two cells again. And let's say that this cell has nothing special, but we want to measure the diffusion of particle X from the outside to the inside of the cell. But this cell has specific transporters that allow or that move particle X specifically. So particle X now can be moved through. The membrane is more permeable to X because it can move through the transporters as well as through the membrane, whereas over here it can only go through the membrane. And therefore the membrane is less permeable to X here than this membrane is. So these 5 things here are things that affect the rate of diffusion. We'll call them factors that affect the rate of diffusion. Okay, and that happens in any scenario. So where there are changes in temperature, concentration difference, molecular weight, membrane surface area, or permeability, we are going to see differences in the rate of diffusion. Now, I want to talk next about a particular kind or a special kind of diffusion known as osmosis. Okay, osmosis is defined as the diffusion of water across a membrane. Okay, so it's still diffusion, it's just the particular case of diffusion of water. So let's set up our scenario again. Our container, which we have split into, and the divider is permeable only to water. Okay, so now we're going to fill our chamber with water. And we're going to dissolve some solutes on each side. Now, I'm not going to draw circles anymore. I'm just going to continue to use numbers here. Let's do percent. So let's say that on this side I have 50% salt. We'll make it NaCl. And on this side I have 25% salt, NaCl. Okay. Now, if this boundary, if this membrane in between allowed sodium chloride to cross, then we would expect the sodium chloride to go from high to low concentration. But this membrane only lets water cross, so the salt is going to stay exactly where we put it. But the water is going to move. Now, how do we figure out what the concentration of water is? We don't usually talk about things, water having a concentration. unless we say that's pure water, which would be 100%, right? So if I have a beaker of pure water over here, then this would be 100% water. Let's say that it's just a matter of simple math and I can figure out what the percentage of water is by looking at the percentage of sodium chloride. So if this is 50% sodium chloride, this will be 50% water. And if this is 25% sodium chloride, then it will be 75% water. So now we can see which side has the higher concentration of water. And water, just like any particle, is going to obey the law of diffusion and is going to move from where water is more concentrated to where water is less concentrated. And in fact, what will happen is that the water in this side will go down and the water in this side will go up because the water is moving from one side to another. Now, it can be quite challenging to work backwards and figure out what the concentration of water is. So one quick way that we describe the osmosis or the diffusion of water across the membrane is we say that water always follows the solutes. So whichever side has the most solutes or the most stuff, that is the side that the water is going to move to. Let's take a look at this again. Okay, so we'll put our chamber back up here. Our membrane that's only permeable to water. We'll fill each side with water. And now I'm going to add some solutes. Use a different color here. So on this side, I'm going to put 60% sucrose. And on this side, I'm going to put 20% sucrose. All right. Now, without going back to figure out how much water or what the percentage of water is in this, if I tell you that water is allowed to move but the sucrose isn't, then you just need to figure out which side has the most sucrose or the most solutes, and that's this side. So that means that water will move to the side that has the most solutes. And the water level on this side will rise and the water on that side will fall. Okay? All right, so that's osmosis. Now let's talk about what happens with osmosis in cells. Typically, cells are able to control their volume by moving solutes from one side of the membrane to another. But in the case of red blood cells aren't very good at controlling their volume. So they become a very good model for showing what happens when you put cells in solutions that have a different amount of solutes than what the cell has. So what I'm talking about now is called tonicity. The tonicity. And tonicity refers to the concentration of solutes in a solution and what it does to cell volume. Okay, let me show you what I mean. Let's take 3 beakers. And in each beaker, I'm going to put a solution. And and a certain number of solutes. Okay, I'm using lots of different colors here. So in this one, let's say I make this one 75% stuff, because it doesn't really even matter what the solutes are. And over here, let's say that I make it 25% stuff. And over here, I make it 5% stuff. All the same stuff in all three, but this one is much more concentrated, and this one is much less concentrated. Now I'm going to put some red blood cells in here, in each of these beakers, or one giant red blood cell because it's easier to draw. And as it turns out, making this up, but let's say that red blood cells have 25% stuff on the inside. So each one of these has 25% stuff. All right, now water can cross the membrane of red blood cells. Other cells have ways to prevent that. The red blood cells do not. So if we look in this scenario, water is in the cell, water is outside of the cell, and it's going to move towards the side with the most solutes, which means that water is going to leave the cell and go into the beaker. That means that the cell is going to shrink. And when a solution causes the cell to shrink, we say that it is a hypertonic solution. Hyper because it has too much stuff in it, and that's what causes the cell to shrink. Now, if we look over here at this scenario where we have a solution that has 5% stuff and the cell has 25% stuff inside, then the water, which can cross the membrane freely, is going to move from where there is low concentration of solutes to where there are more solutes. So it will move into the cell. This results in the cell swelling. And whenever we have a cell that swells, we say that it is in a hypotonic solution. Hypo means low. So it has a low volume or a low concentration of solutes. Now in this scenario where 25% stuff is in the solution and 25% stuff is in the cells, water still moves freely across the membrane, but it moves in and out at the same rate. Therefore, the cell does not change size or shape, doesn't change volume. So here the cell stays the same size, and we say that this solution is isotonic. Iso means same. So the cell has the same volume. So that's tonicity. All right. What happens over here if this continues is the cell will rupture and burst. And what happens over here is the cell shrinks so much it's no longer able to function. So whenever we work with cells in science, we always make sure, unless we're testing the tonicity, we always make sure to use isotonic solutions so that it doesn't change the cell's volume. Now let's look at a cell that actually has a way to adjust its volume. Let's get a nice big beaker here. I'll keep dropping my markers. All right, and it's filled with a solution. And inside, I'm going to go ahead and make it a red blood cell again. All right, so we know that red blood cells have 25% solute inside, and because I'm not paying attention, I'm going to accidentally make this solution, hypotonic. So I'm going to put only 5% stuff in here. Now, just like we saw in the example, if there are more solutes in the cell than there are in the solution, it means that water is going to go into the cell and the cell is going to start swelling. The response of a cell under these circumstances would be to take something like solutes, let's say sodium chloride. I know that's so tiny you can't possibly see it, but the cell will take sodium chloride and move it to the outside of the cell, and that means, because water always follows solutes, that some of these water molecules will also leave the cell, and therefore the cell will not swell or won't swell as much. Okay, now let's look at some membrane transporters. Okay, now we're going to talk about the different ways that particles or substances can get across the membrane. And for that, I need one giant membrane. So here's our phospholipid bilayer. And as always, this is the outside and this is the inside of the cell. And the first type of, or the two types of transport are described in terms of whether they require energy or not. So we have what's called passive transport, and that means no energy. And we have active transport, which requires energy. Okay? I would write it out, but my board's very small. So let's take a look at some things that use passive transport first. Now, inside the cell, we have a low oxygen concentration and a high CO2 concentration, because cells take oxygen, use it up, and produce CO2. Outside of the cell, where it's being delivered by the blood, we have high oxygen concentration and low carbon dioxide. Both of these substances, oxygen and carbon dioxide, simply diffuse from high to low, through the membrane as if the membrane didn't even exist. That's because these are small lipophilic molecules. Right? Because they are small, they can more easily diffuse. Remember, molecular weight affects the rate of diffusion. And because they are lipophilic or lipid-loving, when they get into the middle of the phospholipid bilayer where the fatty acid tails are, the fatty acid tails don't mind them being there, so they can pass right through. All right, so both of them are moving from high concentration to low, from high to low. That means that it requires no energy. It's simply obeying the law of diffusion. And it goes through the membrane and doesn't require any sort of transporter. So we call this type of transport simple diffusion. Now, we also use diffusion for some particles that are not small and lipophilic. Say, for instance, glucose or sodium or potassium, those charged ions. Those, because of the charge, they can't pass through the phospholipid bilayer. The fatty acids are immediately like, can't come this way. We only like lipophilic molecules. So when we want to allow diffusion of something or something to move from high to low concentration, but we don't have, or that substance can't move through the membrane, we use something called a channel, or in some cases, a transporter, like we saw in the previous lecture. Both of these, as long as they are helping something to move from high concentration, let's say we have high sodium out here, and we also have high glucose. And inside we have low sodium and low glucose concentration. The channels will allow sodium to come through to go from high to low concentration. So that's diffusion. And the glucose will bind there and then come out, going from high concentration to low concentration. So that's also diffusion. But what's different is that the diffusion in this case requires the help of a membrane protein. And so we call this facilitated diffusion. Okay? To facilitate means to help. And in facilitated diffusion, just like in simple diffusion, particles are moving from high concentration to low concentration. It actually does not matter that they're moving from the outside to the inside. The same thing could be true if a transporter or a channel were moving something from the inside to the outside. The point is that it goes from high to low and therefore is passive, requires no energy. So these are both forms of diffusion. And as I mentioned, the transporters that are used here, let me get another color. So this would be a transporter or a carrier, and this would be a channel. So those are the two types of membrane proteins that you'll see in facilitated diffusion. Now, I'm going to show you another type of transport in a moment that also uses carriers but is not facilitated diffusion. Channels are always facilitated diffusion. They cannot move something against its concentration gradient. But transporters or carriers sometimes can. So you can't just say, every time I see this, it's facilitated diffusion, because you'll be wrong. Okay, I kind of ran out of space here, but let me put, let's put our active transport in. I'm going to erase this. Okay, so now I'm going to talk about the types of active transport. Put our membrane back. Okay, so in active transport, we're going to be moving things from low to high concentration, which is breaking the law of diffusion, and that's the reason that we need energy. It requires energy to break the law here. So the first kind of transporter is one called a pump, and it's special because it uses a molecule of ATP in order to move substances from one side of the membrane to another. All right, so this is called a pump. And its type of transport is called direct or primary active transport. Now, what this molecule does, this is a specific example of a pump, one of the most important ones, and we'll be seeing this one again. But it moves 3 potassium ions into the cell and 2 sodium ions out of the cell each time it uses a molecule of ATP. So the potassium and the sodium will each bind to this transporter, just the way we saw it bind to the transporters before in that little specific pocket. But the transporter isn't going to do anything until a molecule of ATP comes along and drops off some energy. Okay? So once that energy is here, now this transporter can change its shape and can move the potassium in and the sodium out. Now, how do we know that why it requires energy? That's because inside the cell we have a low sodium concentration and we have a high sodium concentration outside of the cell. And inside the cell we have a high potassium concentration and we have a low potassium concentration outside of the cell. So in both of these cases, the pump is moving sodium from low to high concentration and is moving potassium from low to high concentration. It's breaking the rules of diffusion, and therefore it requires the use of energy, and it's using it directly. It takes the ATP, takes the energy out of the ATP, and then pumps the sodium and the potassium. Now, not all active transport requires a pump. So we also have transporters just like we saw before. These do not require ATP, but they move specific substances from one side of the cell to the other. Now, there's one that transports glucose and sodium, both in the same direction. So it takes glucose and sodium, and it moves them to the inside of the cell. Now, sodium, remember, is at a high concentration outside of the cell and low inside the cell. So that part's fine. That's not breaking the law of diffusion at all. It's moving from high to low. But glucose is moving from low concentration to high concentration. All right, so that one is breaking the law of diffusion. So that means that this is active transport, but it requires energy, but it's not clear looking at this where that energy is coming from until you look back at the pump. So the pump, as you recall, was taking sodium from low concentration and pumping it to high concentration, creating this high concentration of sodium. Now that's like energy stored, right? If we put all that sodium out there, then the more we put out there, the more that sodium wants to come right back into the cell. And that's exactly what happens. This transporter says, hey, sodium, I really want to bring that glucose in, but it does not want to come. It is kicking and screaming. So could you do me a favor and just, hop on? And the fact that you're going down your concentration gradient will allow me to bring the glucose in. It's kind of like, I don't do this, but it's kind of like if you go to a club and you bring somebody along who can get you in, your friend who is extra gorgeous and has the fancy outfit. The bouncer is going to say, okay, she can come, and her friends too. So it's the same thing here. Glucose is allowed to, or this transporter is able to bring glucose in because sodium is rushing through to get back to where it's at a low concentration. So this is called indirect or secondary active transport. And that's because the energy for this comes from the sodium being pumped out and using this ATP, and then that sodium can come back in and bring in a glucose. Probably seems like this is a waste of energy in some ways because we're pumping the sodium out just to let it back in, but we're letting it back in only when it brings along something that the cell actually needs. So sodium is actually just being used to provide the energy, the indirect energy, to bring in something else. And that's indirect active transport. Okay, I think that is the end of membrane transport.